Welcome to the AI Brothers podcast. We are two brothers who have been discussing technology since we were kids and have decided to include you in these conversations.
We will cut through the noise to bring you data and research on the latest developments on AI. We are not interested in hype. We bring you the signal to keep you on top of this new technological and industrial revolution.
Because AI will permeate everything and be everywhere. Initially as a layer on top of existing systems, and then becoming itself the operating system.
Today we will talk about a topic close to our heart: AI agents in biomedical research and drug discovery.
Yes, it is indeed a topic very close to our heart. AI curing all diseases and putting me and all my colleagues out of work.
You'll get to podcaster full time. That's all.
Yeah. I guess that's going to be my future plan once I run out of things to work on in my day jobs. But jokes aside, so I think the reason why we picked this topic today was based on a quote from Eric Schmidt. For people who don't know Eric Schmidt is the former CEO of Google. He's actually the first professional CEO after the foundation of Larry Page, yeah. Yeah, after Larry Page
created Google.
Yeah, so he was the actual first professional CEO, and he said about AI that AI will be to biology what math is to physics, in that it would provide the essential tools and framework to make sense of biology.
Well, before we dive into the paper, let's talk about AI in biomedical research. Where are we?
Okay, so the field of using AI in biomedical research is really a field of intense work. So basically, you have two big families: foundation models and scientific agents. In my opinion, the most advanced aspect is happening in the foundation models. So these are neural networks that have been trained on specialized types of data that is specific for biology. It could be proteins, it could be chemical structures that goes into drug, it could be pathology or radiology scans. The most famous of these foundation models, and I think a lot of people heard of, is AlphaFold, that led to the Nobel Prize.
When DeepMind established the crystal structure of all protein, right?
Well, I mean, did they? I don't know. There was definitely a bit of PR and a bit of overstatement. Not all proteins had their structure identified, because a lot of proteins are very unstructured, very difficult to define the structure of these proteins. But however, it is definitely one of the most powerful tools that have been discovered probably in the past decade, and opened the door to an explosion of what's called protein models. So these protein foundation models, allow you to generate proteins from scratch. These proteins could lead to new therapeutics like antibodies or small protein therapeutics, and have also opened up the door to generate these models, modify the binding of a drug to a target protein of interest, and that opened up tremendous possibilities in drug discovery. So definitely something very important.
Well, interesting. But what about scientific agents? The kind of agents that would actually do novel science in a broad sense, can formulate hypotheses, analyze data, and come up with novel conclusions. What about them?
Well, really about these scientific agents, in a way, these tools, machine learning tools, have always been, in a way, biology was one of the first adopters of these technologies just because of the amount of data that gets generated. But a true agent that can really, as you said, formulate hypotheses, analyze the data, come up with a conclusion, really never got developed. You could have, at the very beginning, you have these, they were called AI models that can do scientific literature searches and synthesis and try to identify new patterns in this research. But again, it was just focused on scientific literature. More recently, you had other agents, for instance, one developed by MIT that can design experiment and then control the execution of these experiments in a fully automated lab.
Well, that sounds really, really incredible, huh?
It is, and the MIT agent did had some impressive result, but it was really focused on chemistry. And again, the idea of automating an entire lab with an agent is still not there. There are a lot of experiments in biology that tend to be quite messy, tricky to set up. So I don't think you'll be able to automate all of this. More recently, there has been the shift to this end-to-end scientist to start from a hypothesis, analyze the data, and go all the way to conclusion. Google, Media, a number of other companies have been trying to push in that direction. But again, it still never felt like we are completely there.
Okay, but today's paper feels different, right? The authors talk about an AI scientist. This sounds like a really big deal, right?
Yeah, it does. So let's tell the people what are we going to talk about today. So we're going to talk about a paper that has been published by Future House, and it's titled Kosmos, an AI scientist for autonomous discovery. And it's currently on archive for anybody to go through it and read it.
Okay, but before we talk about the data, let's talk about Future House. I believe it's a non-profit organization building AI agent to automate research in biology and other complex sciences, right? It raised, I believe, $250 million. It had, I believe, Eric Schmidt as a backer, right?
Yes. Big names, big money, big ambitions. Actually, in one of their recent press releases, they say that they were planning to achieve sort of scientific agents in 10 years, but actually managed to achieve it in 2 years.
Come on, bro. I sense classic snarky comments, right?
A little bit, but actually, no. I think really when we're going to talk about this paper, it is truly one of the major advances. Now, let's be honest, there's a bit of PR and salesmanship, but it is a true major advance.
You still had to make a comment, right?
I can't help it. I can't help it. I always need to make comments when it comes to these great claims.
I mean, I think it's important stuff. They have built an autonomous agent called Kosmos that can run up to 12 hours, starting from data set and a research objective. It runs parallel cycles of data analysis, literature search, and hypothesis generation before synthesizing discoveries into scientific reports. It's amazing, right?
Yeah, it is, and I think when we'll talk about the results, it's gonna really key advance in their model is their ability to run in parallel data analysis and literature search through different agents. So in parallel, there's one agent's doing discovery, one agent's doing literature search, and there's another agent doing discovery. So they're exploring these different paths of discovery, and underlying all of this, they have a constantly updated, what they call a constantly updated world model. So they're doing analysis, they're looking into the literature, and they're updating their ground truth, as you would say.
The scale, and the scale is really important. They say Kosmos execute an average of 42,000 lines of code across 166 data analysis agents rollouts, and they read 1,500 full-length scientific papers across 36 literature review agents rollout, and that is all within each run,
Yes. Yeah. I mean, look, these are big numbers. They even at one point compare them to what a scientist would be able to do if that scientist had to do all this work. Again, probably I'm the more skeptic voice, and I'm not a huge fan of throwing these metrics around and try to what a machine can do relative to a human. But again, I think the architecture of this agent is impressive to be able to run this execution. Okay, so now let's dig into the applications. So they published 7 studies. I don't think we want to go through all of them. We just going to highlight 3 of these studies, but this is again an important paper, and we definitely recommend. So, in the first study, it's biology experiment. So they gave Kosmos mouse brain metabolomic data, and for the non-biologist listening to this podcast, metabolomic is basically the analysis of all the metabolites that are produced in a specific organ, in this case the brain. So I got to think of it as the cell is living its life, it's consuming nutrient, it's secreting either signaling molecules or product of the production of a normal cell, and all these molecules, whatever goes in and whatever goes out, goes into this field that we called metabolomic. And this can be analyzed all at once, and it can tell you sort of how is the metabolism of a brain changing and reacting to a specific condition. So in this experiment, so they took this mouse metabolomic data where researchers were trying to understand how the body protects neurons during hypothermia. So you can understand that if the body is going through hypothermia, the body wants to protect some of the key organs, in this case, the brain. So they gave this raw data to Kosmos, and what's critical is that the results of this data is actually in a paper, but the paper hasn't been published yet. So Kosmos wasn't trained on these results. And the interesting is that just from this raw data, Kosmos was able to identify the same signaling pathway called what's related to the activation of the nucleotide salvage pathway, and they found that this pathway activation is the one implicated and they did that through these cycles of data analysis, literature searches. So basically, they would analyze, Cosmos would analyze the data, identify a pathway, do the literature search, see if it's actually could be the reason for the protection, and they would go through these cycles. So this is sort of discovery of something that humans have already found, and it's almost like a validation of the power of the system.
Yeah, and in the second study, they looked at solar cell manufacturing with the goal of showing that agents could work beyond biomedical research. The goal of the experiment was to look at how environmental parameters such as temperature, humidity, and solvent pressure affect solar panel manufacturing at the coating and annealing stages. Starting from sensor data, Cosmos identified a linear relationship between the solvent vapor pressure and on a solar cell and the cell performance. Here, along showing that Cosmos can perform discovery similar to humans they showed that Cosmos can be used on non-biomedical data, and claimed that the analysis was more extensive, and again it was a sort of rediscovery to see if Cosmos could find results that humans have already discovered and identified, but have not been published. But the result that have not been published, you know?
Yes. So these two studies, I think, were critical in terms of showing that, from the same data, they can make the same discoveries that humans have done. And I think they made it a point to show that they can go beyond biomedical data, although the 6 other studies are all in the biomedical field. So I don't know if it's a bias of the model or it's the bias of the people working in Future House.
Maybe
but
it's
still,
fine tuning. Yes,
maybe it could be.
some
Yeah,
sort of fine tuning. Okay.
yeah, it could be.
Yes.
Yeah.
So this seems like very impressive stuff. I also liked the conclusion of the paper because it was an honest review of Cosmos limitations. They highlighted that although 85% of statements derived from data analysis were accurate, Cosmos had the tendency to invent unorthodox quantitative metrics in this analysis that will often statistically sound, but can be conceptually really obscure and difficult to interpret. They also highlighted that Cosmos was found to be only 57% accurate in statements that required interpretation of results, likely due to some propensity to conflate statistically significant results with the scientifically viable ones, you know?
Yes. And although they say that these problems and limitation will be improved by future iteration, I always can't help but think, is this a sort of fundamental problem of LLMs? There's actually a recent study from Stanford that showed that LLMs are unable to differentiate between facts and beliefs. So anything that looks or seems sound, particularly on the in a statistical sense, would be considered as truth from an L. But just before we go deeper in the conclusion, I think we'll pick up on the third discovery that was shared by the authors. And in this case, they claim that they made a novel discovery. And this is related to neuronal aging. So in this experiment, they picked mice at different ages, and they took single cell RNA expression. So again, for the non-biologist, there is what's called the central dogma of biology, is that you start from DNA. DNA gets transcribed into RNA, which is sort of the messenger that takes the information. And then from RNA, you get a translation, and then you form all the proteins that you have in the cell. And then the single cell approach is a technology that has been around for, maybe a decade or so now. That allows you to, if you take, let's say, a brain, you split it into single cells and analyze the expression in every cell. And the reason why you want to do that is that in the brain, you have different types of cells. You have neurons, but you have glia, astrocyte, you have different types of cells, and every neurons tend to be different neurons. You have dopaminergic neurons, you have glutamatergic neurons, so you have different types of neurons. So you want to really see the expression in every type of cell in that brain. So they took this single cell RNA expression data from mice and analyzed the process of neuronal aging. And from this analysis, they actually identified a novel family of proteins called flippase that have been downregulated. And this leads to what they call an increase of the eat me signal that lead to neuronal destruction. And the data from these from this analysis, the results from this analysis were actually further validated from clinical data, and then they showed that a similar trend was observed. So in this case, they claim that it's actually a novel discovery, that this this neuronal agent behavior was not identified by the researchers who did the analysis of the data in the first place. So
Okay. Well,
maybe
well,
now we'll go back to the conclusion on the what. And to be honest, they actually listed a number of the limitations of this model, and I think this is sort of the reason why we started this podcast series, is that we are able to share with people the actual science and the actual data, so that they're able to make an informed analysis of the state of the AI. And when you really look at the science, actually, the scientists are quite honest with how the discoveries are going. So in this case, they actually talk about it and say that, the fact that the analysis starts and stays in this closed loop system for a long time is not ideal for your typical scientist. Scientists want to be able to interact with the model as it's reason. And I actually tried Kosmos on the website, on Edison Scientific website, and it is impressive. I was able to get it to do quite impressive analysis, but it would be very nice to have a conversation with this model, just constantly going back and forth with it.
the issue of the size of the data set is also something that we need to highlight. 5 gigabytes seems small for biology. I hope this is just a compute issue and not more a fundamental issue with the model,
Yeah, that's, as you said, it's 5 gigabyte in biology is not a huge data set, and I hope, again, as you said, it's a compute, and also I don't know how much it costs to run all these agents in parallel. So the compute cost is also an issue to talk about.
Yeah. Okay. Now let's talk about the consequences of this work, because it is a step up relative to the state of art, right?
Yes. I honestly played a little bit with Cosmos on the website. They only give you 10 credit, but I played with it for over. And honestly, it feels transformative. If they actually manage to fix the limitation that they themselves stated on the website, it would be a very, very powerful tool.
You don't seem to think it will replace actual scientists, right?
I don't think so. Because I don't think you'll be able to completely replace and automate science. So on one hand, you have the acquisition of data, and anybody who's been into a biology lab will tell you that it's sometimes quite messy, and the most important discoveries are still about optimizing a technique that is very hard to automate. There's probably going to be automation of, in drug discovery, and it's already been done, but not for the lead in discoveries. And then I think for the top science, there is a question of taste. And the best scientists have taste for good questions. And that is hard to develop. Even top, well-trained scientists take a long time to develop this taste for good question. And I think it will take more time for these agents to get there. However, as a tool, I think it is absolutely super powerful.
So I think so your perspective is that this AI is just automating tasks and not a full AI agent. I think it's what your perspective is quite similar to the perspective of Eric Schmidt himself, one of the backer of this project, I believe. He said that right now AI agents are middle to middle. They're not end to end. They don't have a non-stationary objective function. They can't really go through a problem from beginning to end. And even although with all the deep thinking, they're just not able to go through a problem from beginning to end. They just go through middle to middle.
It's
Because they don't have a non-stationary objective function. But
it's
people do have this what we call a non-stationary objective function.
it's music to my ears. I'm in good company. If Eric Schmidt and I think the same, I think I'm in good
Yeah.
company. So this is very good to know. This is music to my ears. I think also the other thing is, if we look at at least that's how I look at it. If I look at AI agent as just these tools of automating tasks and accelerating tasks, it becomes clear that the most important skill that we should acquire as human is the sense of agency, is the willingness to do things. And I remember a long time ago, I watched this video from Jacques Brel, the Belgian singer, but he's very famous in French-speaking world.
I do remember him.
Anybody who's in the, of course we do remember. And he said something. He said, "Le talent, c'est avoir l'envie de faire les choses." In English, it is talent is the desire to do things. And at the time, it was probably, I don't know, decades ago, it seems very corny and cheesy thing to say. But I think now that we have these agents that automate so much of what we do, the sense of agency, the willingness to do things is the true expression of a form of talent. So I think in a way, old quotes are very good to ponder.
Yeah, I think we can close the podcast on this last classic note. Thank you for listening. I hope you enjoyed it, and please leave comments below and share if you think your friends will enjoy this video.
Thank you very much.